morning, I am Madhurima Datta, Zoology and Biology educational content creator. Now's content for B.Sc Zoology Honours, Microbiology Honours, Physiology Honours and Biochemistry Honours, and also CUT PG exam, GATE exam, DBT exam, IIT JAM exam and CSI NET Life Sciences exam. Paper Cell Biology, Unit 1, Introduction of the Cell Biology, and topic is the difference between prokaryotic cell and eukaryotic cell. Let's go. The greatest biochemical diversity exists among prokaryotic cells. So I will discuss about the short introduction of the prokaryotic cells. From simple microscopy, it has long been clear that living organism can be classified on the basis of the cell structures into two groups, the eukaryotes and prokaryotes. Eukaryotes keep their DNA in a distinct membrane-enclosed intracellular compartment called the nucleus. The name is from the Greek meaning truly nucleated. From the words eu means well or truly and karyon or kernel means nucleus. Most prokaryotic cells are small and simple in outward appearance, and they live mostly as independent individuals or loosely organized communities rather than as multicellular organisms. They are typically spherical or rod-shaped and measure a few micrometers in linear dimension. They often have a tough protective coat called a cell wall, beneath which a plasma membrane encloses a single cytoplasmic compartment containing DNA, RNA, protein and many small molecules which is needed for life. Next, difference between prokaryote and eukaryotic cells. Example of the prokaryotic cells, bacteria and archaea, and eukaryotic cells examples are protist, fungi, plants, animals. First features, nuclear architecture. In prokaryotic cells, no true nucleus, genomic DNA resides in an irregular, non-membrane bound region called the nucleate. And in eukaryotic cells, true nucleus enclosed by a double membrane nuclear envelope embedded with the nuclear pore complex or NPCS. Next features, genomic DNA topology. Typically, a single circular double-stranded DNA molecule, that is plasmids, that is extrachromosomal, are common. And in eukaryotic cells, multiple linear double-stranded chromosomes complexed with histone octamer to form chromatin. Next features, transcription and translation. In prokaryotic cell, transcription and translation process are coupled. Means transcription and translations occur simultaneously in the cytoplasm due to lack of spatial compartmentalizations. And in eukaryotic cells, spatially separated, that is transcription occurs in the nucleus, mRNA is processed, and post-transcriptional process, that is capping, splicing and polyadenylation are occurs and then exported to the cytoplasm for the translation. Next features, ribosome sedimentation. In prokaryotic cells, 70S, that is total 40S large subunit and 30S small subunit prokaryotic cells ribosomes are present. And in eukaryotic cell, ribosomes are found as 80S, that is 60S large subunit and 40S small subunit. Organelles like mitochondria, chloroplast contain 70S ribosomes. Next features, cellular biochemistry. In prokaryotic cells, that is a best example of in archaea, ether-linked branched isoprenoid, that is often forming monolayers and lacks of sterol except mycoplasma which use hapanoids. Next, in eukaryotic cells, Ester-linked straight-chain fatty acids contain sterols, that is cholesterol in animals, ergosterol in fungi, phytosterol in plants for fluidity in regulation. Next feature, cell wall composition. In prokaryotic cells, in bacteria, peptidoglycan, murin alternating NAM and NAG units are present, and in archaea, pseudopeptidoglycan or S layers or polysaccharides are present. And in eukaryotic cells in plants contain cellulose and hemicellulose, fungi contains chitin cell wall, and animals contain surrounded by extracellular matrix. Next feature, cytoskeleton elements. In prokaryotic cells, primitively homologous FTAZ, MRE and MBL are present, and in eukaryotic cell, microtubules, microfilaments and intermediate filaments are present. Cell division mechanism. In prokaryotic cells, binary fissions are seen. Binary fissions driven by the assembly of the FTZ ring, and mitotic spindle apparatus is completely absent. And in eukaryotic cell, mitosis and meiosis utilizing a microtubule mediated mitotic spindle apparatus. Now the topic has ended. Please subscribe my YouTube channel Biogenesis by Madhurima and follow my Facebook account Madhurima Datta for all video of graduation, master's, CUET, PG exam, CSNET, Life Sciences exam, GATE exam and IIT JAM exams. And follow my Facebook account Madhurima Datta. Thank you everyone.